So today we'll do something a little different, which is we'll begin the lecture by discussing in some detail the paper that you were reading for the quiz, so it should be familiar to you. So the goal of today's lecture is to spend about, I want to say, a third of it talking about that Vaccine X paper, and specifically the T cell versus B cell considerations. And then we'll switch over to talking the genetic basis of human disease. So this course is not going to be, 100% about COVID. So I need to give you some background in order to get back to COVID related articles, which is, the next sort of major topic is trying to figure out why do different people respond differently to this virus, in addition to just the pure immune response. All right. So last time we discussed B cell immunity and went over a recent Nature review article where the current efforts in vaccine design were discussed. So we talked about the different ways to design immunogens, the different way to deliver them, different way to evaluate their efficacy, and a little bit about the regulatory process. Most of the vaccines that you've likely received as a child or as an adult were designed to create antibodies, which can neutralize viral particles. So they're B cell centric. And if you recall from the review that we had discussed, that most current SARS-CoV-2 vaccines aim to raise immunoglobulin G antibodies. And if you remember, there was a distinction made between IgA, which provides upper respiratory tract protection, and IgG for the rest of the genome. Question is, will this lecture be posted on Canvas in PDF format? I thought I did. Oh, sorry. I'll repost it as a PDF file. I uploaded the Keynote source, but well, we'll sort it out. The issue with sort of focusing too much effort on just IgG antibodies is there's some reason to believe that this might not actually protect you from infection. If a successful vaccine might protect you from getting the disease in a severe form, so you might become asymptomatic, for instance, but it doesn't necessarily sterilize your system, so to speak, against any possible infection by SARS-CoV-2. And by focusing on spike alone, which a lot of the, especially recombinant vaccines are doing, and some of the mRNA, you might sort of narrow the target that the immune system is seeking to only one part of the viral genome, which may be suboptimal. There is evidence from SARS-CoV-2, other coronaviruses, and other respiratory viruses that B-cell mediated immunity may wane, meaning that your immune system generates appropriate antibodies when you are infected or vaccinated, but they don't last very long. So they kind of decay over time. They don't create a lasting memory response. Question is it possible that multiple competing vaccines would be released? Oh, absolutely. There will be many, many vaccines. I mean, there are 180 in various stages of being trialed. And I'm sure that every company that is rushing to release it to market as quickly as possible, and obviously as safely as possible, will do it. And there's enough differences between the vaccines that they can be justifiably released, because some might be better for certain type of the population, older people, younger people, and so on. But we don't have that information yet. It'll create an interesting situation for us as health services consumers, because we might be inundated with different choices of vaccine. So we'll see how that will be navigated. And there's also, going back to the presentation, there's several lines of evidence that T cell responses, which are usually relegated to secondary considerations for a lot of the vaccine design efforts, they're unusually robust and maybe longer lasting. And in addition, when we talked about this a couple of lectures ago, there were several really beautifully executed papers that demonstrated that people that had never seen SARS-CoV-2 couldn't have seen SARS-CoV-2 because those samples were from 2015, 2016. Their immune system harbored memory T cells that were activated and bound to SARS-CoV-2. So the implication there is because there are other viruses that are related to SARS-CoV-2, other beta coronaviruses that cause common cold. A substantial proportion of the population at some point have been exposed to a similar virus, and because of that, there might be some protective cross-reactive immunity. So with this in mind, can or should these considerations influence vaccine design, which was the subject of this very well written, in my opinion, paper by Mark Hellerstein from UC Berkeley. And sort of the argument that he lays out follows the following sort of succession, as you can convince yourself by looking at the available literature. First, SARS-CoV-2 vaccines will likely be licensed based on neutralizing antibodies in phase 2 trials, right? So this will, and we can talk about that a little bit later. You know, what is the minimum standard for licensing a vaccine? But you know, suppose you have a vaccine that is tolerated, that shows efficacy in, say, phase 3, and it raises neutralizing antibodies to SARS-CoV-2. The question at this point might be: is antibody response in coronavirus infections a sufficient outcome to gauge protective immunity? So, is this vaccine going to deliver a long-lasting protection, which we would like to have? As arguments against this, one could say that B-cell response may be shorter lasting than T-cell response for coronaviruses. You'd have to justify this, but let's just accept this at the moment. There is some evidence that strong B-cell response is correlated with more severe diseases. So this actually might be a double-edged sword. You might have over-eager immune response that complicates disease progression, whereas strong T-cell response is claimed to be correlated with less severe disease, right? So presumably, in this sense, T-cell response is predictive of a less severe disease. And antibody protection may not be sufficient for protection against infection. And data so far point to the significant role of high-quality T-cell responses in protective immunity. And moreover, there were a few successful vaccines raised to other viruses, specifically yellow fever and smallpox, that induce T-cell responses, specifically this particular subset of T cells, long-lived virus-reactive CD8 or cytotoxic cells that are present and measurable within just a few months of vaccination. So they rise up quickly and they last a long time. And if you start looking at which part of the virus is being targeted by the T-cell response, there's some other unusual features about SARS-CoV-2 that are revealed. For instance, whereas B-cell epitopes will be skewed towards spike based on the information that we have. In fact, you look at neutralizing antibody papers that people have published, many groups have published since March, they all are raised against spike. But T-cell epitopes are much less dominated by spike protein in SARS-CoV-2, which means that this branch of your immune system targets the entire genome. And in fact, it doesn't pay particular attention to spike. So most vaccine candidates are focusing on spike protein as antigen. But if you look at what happens during natural infection by SARS-CoV-2, your immune system, if it has to fight the actual infection, induces broad epitope coverage. So it doesn't just focus on spike. So if you design a vaccine that only presents this part of the virus, the corresponding immune response might be inadequate. And it's cross-reactive with other beta coronaviruses. In order to better design long-lasting vaccines, or at least consider the options, we need to understand the relation between breadth, functionality, and durability of T-cell responses and resulting protective immunity. And I'll explain using slides from figures from a couple of papers what breadth, functionality, and durability mean. And there's a lot of there's a lot writing on the success of the first vaccine or several vaccines that are released. For instance, you might imagine a public health and disaster that would occur if the first vaccine was a failed vaccine when deployed to the large population. I mean, there's already considerable distrust of vaccination in general in some subsets of the population, and now with added complications because of political nature, then it might be even more skeptically received. And vaccine design, therefore, should be informed and evaluated not only by B cell responses and antibody protection. This is an important subtle point, which means that even if you don't change the design of the vaccine, it shouldn't be, you shouldn't be able to ignore the T cell response when you evaluate it. So basically, so long as you're doing evaluation of what the vaccine is doing, evaluating what the T cell response looks like, even if the vaccine itself is designed to mostly raise B cell responses, it should be done. There's a question. COVID attacks everyone differently, and as for vaccines, the vaccines will interact differently in each individual. There's an article mentioned that a trial was ongoing and that individual had developed some sort of neurological complications. That is, so there's, after the lecture, I'll post a link into Slack. There was actually a really good and accessible article published in Wired, a technology magazine that discusses a lot of the specific sort of endpoints and safety considerations that different pharmaceutical companies have adopted. But there are minimum federal standards which have to be met in order for the vaccine to receive an FDA license. But in addition to that, pharmaceutical companies and vaccine developers are free to add their own. And there's actually enough variation in how they're doing it. So there was AstraZeneca in the UK, there was a neurological complication. We don't have detailed data on that yet because of patient privacy and so on. But we simply haven't had enough time to do a proper large-scale safety evaluation. If you remember from last lecture, a typical cycle for vaccine development takes a decade, and we're trying to compress this in a year or year and a half. The first question is, let's address this one. Is antibody response a good marker for protective immunity? So for example, if you are doing a vaccine trial, phase 2, so a larger population, a vaccine will often be benchmarked by antibody responses. So, are there neutralizing antibodies or reactive antibodies being raised? Are there good viral titers? And if you remember from the review article, every single one of the vaccines that was evaluated had some sort of a B cell measurement, but very few had T cell measurements. So the article that you guys read made those are pretty convincing, in my view, argument that T cell responses might be a better marker after natural coronavirus infection. And in order to show this, the author goes back to the original SARS-1 infection, which has the benefit of having occurred close to two decades ago, so we can talk about really long-lasting immunity. And in SARS-1, so you had 50% of survivors of severe cases had detectable antibodies after 3 years, right? So all of them had it in the beginning, but after 3 years, 50% of them lost it. And after 6 years, none of the survivors had detectable antibodies. As a contrast, virus-specific T cells lasted between 6 to 17 years, and 17 years is basically up to the present time, so they still have it. So just based on this measurement, T cell immunity seems to outlast B cell immunity. There was also a similar lack of antibody response in MERS, in mild diseases. If you look at other related viruses like common colds, the antibodies died away within 1 year, so that they're not long-lasting in general for coronaviruses. And if you look at sort of specific detailed studies of SARS-CoV-2 infections, if you're asymptomatic, for instance, if you do have an infection, but it's not, it doesn't rise to the clinical level, so you don't really have strong symptoms, you might have something mild. So 40% of those individuals are seronegative for anti-spike IgG, which means that they don't have detectable antibody at 12 weeks post-diagnosis. So implication of this, that a large fraction of people that get SARS-CoV-2 but without symptoms might clear it without ever developing a neutralizing antibody response. So how can we, where do you get this claim, for instance, about acute versus, clinical versus asymptomatic patients? So there was this one paper cited by this review. It was in Nature Medicine, Clinical and Immunological Assessment of Asymptomatic SARS-CoV-2 Infections. So notice they specifically wanted to compare and contrast the differences between symptomatic and asymptomatic as far as viral dynamics go. The take-home message of this paper is quoted here. So increasing evidence has shown that asymptomatic individuals can spread the virus efficiently, and the emergence of these silent spreaders of SARS-CoV-2 has caused difficulties in the control of the epidemic. So here's a figure from the manuscript that is used to argue that from the standpoint of viral replication and viral transmission, there really isn't a whole lot of difference between symptomatic and asymptomatic individuals. So if you look at panel A here, the top figure, so these are box charts or box plots with data points overlaid over them. So you have 37 individuals in the symptomatic group and 37 individuals in the asymptomatic group. What's being measured here is cycle threshold of RT-PCR, qRT-PCR detection. And if you remember, this is a quantitative measurement of how much virus there is per unit blood. And the lower the cycle threshold is, the higher the viral load is. The purpose of a plot like this is to compare whether or not there are statistically significant differences between the two populations. So one hypothesis might be that, say, in asymptomatic individuals, there's a lot less virus than there is in symptomatic individuals. Just raw counts of viral particles. That would be one reasonable explanation for why you have an asymptomatic infection. You just have less virus. And what these plots purport to show that this is not the case. So what they do is they do qRT-PCR on two different genomic fragments. So if you remember, qRT-PCR has probes and primers, so you target it to a specific gene and the virus. So these are two independent measurements. They measure the same thing, but they're just measure different genomic regions. So this is in a way a natural replicate. And what you would do is you would compare. So cycle, for example, if there was a lot less virus in asymptomatic, you would expect the cycle threshold for blue points to be much higher than it is for the pink points. Because remember, higher cycle threshold means lower viral load, right? So and this is logarithmic. So if you go 5 down by this scale, this is basically decreasing the viral load by a factor 32. So this is one question here where you basically say there is no statistical difference in terms of how much virus there is. And the second one is a different way to look at it, which is a time series, so survival curve. So positive rate of viral RNA. And this is meant to tell you, basically measure how long are those individuals infectious. So what you're doing, you're taking a number of measurements over time. So, 0 means this is when the infection occurred. And then as you progress, this is just, say this is a month after the initial symptoms. And if, say, let's follow the orange line. So this is a survival curve. At the beginning, everybody's infectious, 100%. Then at day 10, 10% of those become uninfectious, so you can't detect viral RNA. And as you proceed through time, more and more of the individuals drop out. And by the time you're at month 1, basically none of the symptomatic cases are infectious. What you see here is actually somewhat counterintuitive, because it seems like asymptomatic cases remain infectious longer, right? So you have sort of this blue curve is offset, right? So it decays slower than the red curve, and there's a p-value or statistics associated with it. So, and the hazard ratio is just one way to estimate how more likely it is that an asymptomatic case is more infectious than the symptomatic case. So this is a counterintuitive finding, right? So the first finding, basically, there's no difference between how much virus is being made in symptomatic and asymptomatic individuals. And the second one is that both are infectious, and if anything, asymptomatic cases are infectious longer. All right, let me take the questions. I've heard that SARS-CoV-2 infection has potential for other tissue damage, e.g. heart damage, that hasn't been seen commonly in other coronaviruses. If we believe that antibodies are able to worsen disease severity of SARS-CoV-2. That's a great question. I think in terms of antibody involvement in tissue damage. So the first question is, yes, there have been some reports of myocardial damage, even though some of those papers have been shown to suffer from a number of weaknesses. But there's definitely evidence that something happens in a small fraction of individuals. It's actually difficult to interpret, mostly because we don't actually care that much about other coronaviruses, so we don't study them as intensively. So there's a potential for some other tissue damage that just would not be attributed. Say, if you might get rare tissue damage from common cold viruses, you just would not know this because you would not even know that you had a common cold virus. And as far as antibody involvement, it's possible, but you really have to look at the specific tissue and the specific disease etiology. So at this moment, I would say it's not known if definitively they play a role in such damage one way or the other. So it's a possibility. But in fact, next time we'll discuss a specific example of antibody involvement, not in tissue damage, but in basically killing the efficacy of the immune response against coronavirus infections or viral infections in general. Another question is, does this refer to entirely asymptomatic patients or pre-symptomatic patients who will eventually develop symptoms? No, asymptomatic. When you read this paper, it basically defines what asymptomatic and asymptomatic means. So this will be your traditional acute respiratory syndrome, right? So symptomatic, fever, cough, those are well defined. So if you go to the paper, it'll tell you exactly what this means. This does not refer to people that might develop additional symptoms, after a month of infection. And another question, how can two similar sets of data interpreted differently reveal two different p-values? They're actually different data sets, right? So this is a single time point measurement. This is a different type of analysis. So this basically tells you at a particular time point, do you have differences between viral loads? And this tells you something about the time course of the viral load. But since the p-value was mentioned, it just occurred to me that, historically I found that a fair number of people are confused about what p-values are. And if you are confused, you're not alone. A lot of statisticians are confused about what p-values are, and a lot of doctors are. But I thought I'd just show you on the example of this specific, what does it mean to have a p-value? How do you interpret this result? So for instance, here you have a finding that these two data sets are not significantly different, right? So let me switch over to a different screen. And I'll show you a little visualization tool that I found, that I wrote specifically to demonstrate this. So what you're doing here, it's a very simple simulation-based exercise. So you have two populations, right? So in this case, just keeping in mind the specific example that we had, say you're looking at patients that are symptomatic versus patients that are asymptomatic. But you're just, it could be any kind of different experimental conditions, different ages. So you take two sets of numbers, right? And you have a very simple question to ask. Is the median, which is, the 50th percentile of one distribution, different from the other, right? So what I wrote here is a simple simulator that says, how many data points do you want to generate? So, in that particular case, we had 37, so we can do this. We can type in 37. And say we want to detect, the differences between median values in sample 1 and sample 2. Right, so here's an example of what actually happened. So I specified that there should be no difference between the two samples. So these are, they should both be 1. But there's some noise. There's just some statistical noise that's added into this simulation. And because of that, you will see some measured differences, right? So this is exactly the type of box plot that you saw in the paper, where there's population 1, population 2. We know there should be no differences between them because, you know, that's what we put in. There's median exactly the same between two samples, 1 and 1. But you see that if all you do is just look at them, they're different, right? You know, one is less than the other. And if you roll the dice again and do another simulation, they'll also be never exactly the same, right? Sometimes one is greater than the other, sometimes, right? So there's a lot of statistical noise as you're running these replicates, right? This is basically every time I drag this, I'm generating another set of data. This is just randomly generated by the computer. So in order to tell me, as a statistician, what I want to do, I want to say, I don't get to observe what generated the data. I don't know the truth. What I get to do is observe the data points. And I want to see, are those different or not? And a lot of statistics can be intuitively understood by simply, you know, doing visual comparisons. So, you know, here, these plots don't look too different, right? If I do something like this and reduce the level of noise, you will now see that the two plots are very, very different. So one is a median size of sample 1 is 1. It's 2 now for the second sample, and I've reduced the level of noise, which means that the samples completely are separate, right? So, you know, if you see something like this, your intuition should tell you, yes, these are different samples, right? And I can do this, and I can increase the amount of noise. I can even make it very noisy, right? To the point where you now see that as you add more and more noise, and it could be experimental noise, it's actually harder to separate. But let's go back to you know, this situation where we have median for sample 1 of 1, median of sample 2 for 2. So they should be different. The level of noise is 0.5. And there's a particular statistical test that I'm trying to demonstrate here, which is actually what this paper uses. So it's called the Mann-Whitney-U test. All it does, it takes these numbers, it transforms them into one number called a test statistic. So in this case, this test statistic is 324. And this test statistic somehow should be then interpreted to tell you whether or not these values, these two distributions or two samples come from different median distributions. Right, so let's see, this is, and the actual technical details of how this is done, you can read the Wikipedia article. It's actually quite accessible if you want to see how this is done. This is one of the simpler tests to understand. So you have this test statistic, which tells you about how unlikely this observation is. If you've done any statistics, you might know that there's the standardized way to express your confidence in, or you know, your degree of expectations called a z-score. So z-score is just basically sort of, you know, in terms of, it expresses something in terms of standard deviations away from the mean. And you can convert the z-score into a p-value, right? So in this case, the test statistics of 324 gives you a very significant p-value. So let's see, you know, what happens to this test statistic as we make samples even easier to separate, right? So now we have a median of 1 and 5, so it should be easier to tell them apart. The test statistic went to 8, right? Let's take it to 10, right? The test statistic is now 0, right? So this test statistic is small when the samples are different. And if you set them to be 1, so the same, the test statistic is now big. And you can convert this number into a z-score and a p-value. And a p-value pretty much tells you how likely it is that you will observe a test statistic of this value if there is no difference between the samples. So this is, you know, small p-value means unlikely to observe. So a couple of other intuitive points that I would like to show you is that, you know, as you increase the number of noise, increase the level of noise, things become harder to separate, right? So 1 and 2 with a level noise of 1, you know, gives you, say, a z-score of -2 or p-value of 0.01. And then if you increase the level of noise, now basically you can't tell the samples apart. There's too much noise. All right, let's go back to some simple level of, a smaller level of noise. And then let's say, now we want to tell apart two things that are different, but not very different, right? So median sample of 1 is 1 and then 1.1. So they're different, but they're not very different. And if you look at them, in fact, they don't look different at all, right? And you can sort of keep generating the numbers. And you're going to see, you know, that they don't look visually different. one of the ways to enhance your ability, this situation is called small effect size, where the differences are quite small, right? So they're there, but they're not very easy to find. So one of the ways you can find them is you just increase your sample size, right? So you take, say, 500 points or 1,000 points. So now they don't look very different visually, but there's enough signal from all these data points for the statistical test to pull out a significant p-value. And the other thing that I wanted to show you is that even if you have no significance, right? So the test, we generate data to be exactly the same with noise. If you do enough random replicates, I'm just going to scroll this until I find something that is. So here we go, right? So we just generated one random sample from data points that are actually, have no difference in the medians, but because of the noise and the statistical variation, this particular sample has a p-value of 0.04, which would be reported as significant. So sometimes you can get significant results randomly, but it shouldn't happen too often. All right, so that's so I was going to say about p-values. Now let me go back to the other presentation. So this figure and those p-values were actually exactly derived with the Matt-Whitney U test. This is a different kind of test, but fundamentally it's all the same. It takes these data, transforms them into some test statistic, which goes, either high or low, depending on whether or not the samples are different. It converts it into a p-value, which is basically a measure of statistical significance. So how likely would you be to observe this if there were no differences? So p-value of 0.03, for example, means that if there were no differences between symptomatic and asymptomatic cases, you would have to run this experiment 30 times to get one that is just by chance, has this pattern or a more extreme pattern. So going on to the second part of the paper. So the first paper said that the first part of this analysis said there's no difference between symptomatic and asymptomatic in terms of how much virus they make or how transmissible those viruses are. That's a question. I'll come back to it in a second. And the take-home message for this set of figures is that these data suggest that asymptomatic individuals had a weaker immune response to SARS-CoV-2 infection. The reduction in IgG and neutralizing antibody levels in the early convalescent phase might have implications for immunity strategy and serological surveys. So this was the paper cited by the review, and they said 40% of the asymptomatic cases have no immunoglobulin response after 2 weeks. So this is panel E of the figure. So let's look at this first. What they do is they measure whether or not there's IgG response to SARS-CoV-2 in either symptomatic or asymptomatic patients, either in the acute phase, which means early in the infection, or convalescent phase, which I think is defined as 12 weeks post-infection. And you can see by these pie charts that in the acute phase, about 80% of either symptomatic or asymptomatic individuals develop antibody response while the infection is ongoing. But if you look at what's in their blood after the infection has been cleared, 87% of those that were symptomatic have remaining antibody responses. But only 60% of those that are asymptomatic have those antibody responses. And the reason this is connected to the vaccine is you can think of any successful vaccine, at worst, you should have very, very mild symptoms from it, right? If it's an inactivated or attenuated virus. So what a vaccine would do would be very similar to having an infection that is asymptomatic, or even less mild, pronounced than that if you don't have a replicating virus. So you can take these data to say if you give somebody a vaccine that has similar immune profile as an asymptomatic infection, then based on these data, 40% of them are not going to have any antibodies in a couple of weeks' time, right? That's one way to interpret these data. And these are just, more detailed ways to look at what happens here. So you have more, this is total antibody in asymptomatic and symptomatic, and you can see that there's a significant p-value now between them. Right, so there's more antibody in symptomatic patients, and there's more immunoglobulin in symptomatic patients. And this is a decay curve, which is just matched values where you have how much immunoglobulin there was between acute phase and early phase. So how quickly do antibodies decay? And here they all decay pretty quickly. And this is here the level of detection. So none of them actually, well, maybe this guy, there are a couple that went up, but by and large, the level of immunoglobulin and neutralization activity decays pretty quickly. Question, a lot of viruses have been involved in initiating or triggering an autoimmune disease. Additionally, COVID has been linked to potentially autoimmune diseases like Kawasaki. Do you think it's likely the vaccine for COVID? I don't think it's, I mean, anything I would tell you about a vaccine would be pure speculation, right? Because you would have to look at what the vaccine is, you know, what type of population is being deployed in. And if you look at a large enough sample of people, I mean, if you give a vaccine to a billion people, you're going to see very unusual outcomes just because you have a billion different outcomes. You know, there's no evidence at the moment that this would be a significant concern. Generally, and then this is just generally because we have a lot of exposure to other vaccines, and there's no conceptual difference other than, say, mRNA vaccines, which I haven't tried. But if you're looking at recombinant vaccines or inactivated pathogens, all we're doing with SARS-CoV-2 is putting in a different gene. So if you had systemic issues with autoimmune diseases, you would have seen it with other vaccines. So, you know, rare cases, possibly; widespread, very, very unlikely. So moving on to the next possible consideration for antibodies, is antibody response a good marker for disease outcome? Right, so maybe it's not a good thing to have too much antibody. This part of the review is not as strong as the others. So there's some, but in my opinion, not great evidence that strong antibody response is correlated with more severe disease, while strong T cell response is correlated with less severe disease. A lot of it is based on small samples and limited studies, but you know, here's one of the lines of evidence here. So MERS survivors with longer hospital stays and higher ventilator support had higher antibody level, and there was opposite pattern for higher virus-specific T cell counts. Right? It's difficult to interpret. Right? Association does not mean causation. We'll come back to this later in the lecture. There was also higher IgG levels versus spike in SARS patients who died, and also worse clinical lung injury. And this is original SARS, right? So we're not talking about SARS-CoV-2. In SARS-CoV-2, having a poor T cell response is a predictor for high risk of death. An antibody response appears higher in severe disease. And sort of the most convincing part of me is that virus is not abruptly eliminated after antibodies appear. So it's not a magic bullet. Once the antibodies are detected, they're not so strong that they clear the virus immediately. So it takes a while. So here's an example of a study that was cited in support of differences between levels of immunoglobulins between critical and subcritical cases. So here's the paper. The take-home message is high sensitivity assays with a robust dynamic range provide a comprehensive picture of host antibody response to SARS-CoV-2, IgM, which is early, and IgG, which is later. Responses were significantly higher in patients with severe than mild disease. These differences may affect strategies for seroprevalence studies, therapeutics, and vaccine development, right? So those are the key messages. Basically, there's more immunoglobulins, more antibody response in patients that have a severe disease. So to understand what they studied, here's an example of a pretty nice graphical description of the primary cohort, which they looked at. So there are 94 patients that came in. There were 32 outpatients, and they were all put into the non-severe category or mild disease. And then there were 62 inpatients, out of which 26 went to ICU, and then 36 went to non-ICU. So basically it's ICU versus non-ICU, and you had a convalescent cohort which recovered, and they were also not involved in ICU. The picture that they have here is, again, a sort of kind of an interesting dot plot with ranges, which shows you a couple of things. If you look at panel A, this is a quantitative readout of how much IgM So early immune response, antibody response is there against SARS-CoV-2 in some arbitrary units. And what you would basically want to do is you want to look at non-ICU and ICU. So these are two categories, mild versus severe, and then what stage of the infection it is. So you can basically, in order to compare apples to apples, you would take non-ICU 1 to 5, ICU 1 to 5, and see that they're different. So it doesn't seem there's a whole lot going on early, but then once you get to say 16-20, so 3 weeks into infection, non-ICU is basically to where it was in the beginning. But in the ICU patients, there's a much, much higher level of IgM. And the same for IgG, except you would expect these dynamics to be sort of delayed because IgG comes up later than IgM. So there's really no interpretation here. All you see is an association. So we don't know why it is. I mean, it could be that you need antibodies, that the severe disease simply causes a higher level of antibody. There might be no feedback, but it's just an association. We can't say that you have severe disease because you have an immune response. It just, they both seem to be correlated. So the question is, can antibodies sometimes make things worse? Specifically, can there be this issue of antibody-dependent enhancement? So at the moment, there's no evidence from SARS-CoV-2. There is some evidence from other diseases, and we should definitely look out for it. So there are evidence from animal studies that suboptimal antibody response can sometimes make infection worse. So the 3 examples cited here would be in cats. So in feline, infectious peritonitis, which is also coronavirus-caused disease, worse severity following vaccination or passive immunization with serum from cats that had it and recovered. In ferrets, SARS-CoV-1 virus causes hepatitis only if you had previous vaccination. In macaques, immunoglobulin response against spike worsened SARS-CoV-1 induced lung damage and cytokines. There's definitely cases in other human diseases, for example, dengue hemorrhagic fever, which only occurs if you have an initial infection and antibody response. So the secondary infection is enhanced by the primary infection. And some of the mechanisms of enhancements, this is also really vague, and you have to look at very case-specific details to understand what's going on. But intuitively, what happens, you have antibodies that bind to the virus but don't neutralize it. So if they bind to the virus, sometimes it can actually promote viral entry into cells, including entry into an activation of macrophages, right? So you can basically have a virus that's not neutralized, but is now absorbed into macrophages, and it's still able to do its thing. So it can also occur through low affinity antibodies, so the ones that bind but then detach. You can have cross-reactive antibodies to different strains, so they bind to SARS-CoV-2, but only weakly because they recognize some epitopes that are not particularly conserved. Or you can simply have too few neutralizing antibodies. All right, so here's an example of another study of antibody response to SARS-CoV-2 in patients with novel coronavirus disease. So this is from a Chinese cohort. So the take-home message from this, it should be noted the increase in antibodies were not always accompanied by RNA clearance, particularly in the 3 critical patients. So this is now implied that simply the presence of antibodies does not lead to clinical improvement. So it's not enough to clear the infection, at least in some cases. So this finding suggests that antibodies may not be sufficient to clear the virus. In the pooled analyses of all involved patients, the average antibody levels showed a marked increase at approximately 1 week after the onset and were continuously elevated during the next 2 weeks. Further analyses suggested there was no significant differences in antibody tests between critical and non-critical patients early on, before day 12. However, then they do a subsequent analysis using the statistical technique called general estimating equations. That you can basically throw a bunch of things in and see, do they influence severity of illness? So what they did and they found is that basically what you're trying to say, what is it that is shared by individuals that develop a severe disease and is different from those that have mild disease? And what they found are the couple of things that we already know. So age, so higher age is associated with severity of outcome. Gender, so men are more likely to have a more severe outcome than women, although not by a giant margin, but it's still notable. And independently of this antibody titer, so the higher your antibody titer is, the more likely you are with the statistical significance to have a severe outcome. So you can look at the profiles here. So this is the quantitative plot, so readout C, which basically tracks the amount of antibody that you have. And there's not a whole lot of difference early on, but then in critical patients after day 12, your antibody levels take off. All right, so to summarize this, there's some evidence. It's not, it's not based on very large samples, so it might not hold up to replication, but there seems to be a couple of things that we can take away from it. One is that having more antibodies does not result in better clinical outcomes. It doesn't mean that having more antibodies causes poor outcomes. It's just that they're associated. So it could actually be, a marker of something else. How does cross-reactive antibodies result in enhancement? So cross-reactive in this sense means that they were raised to some other virus and they weakly bind to SARS-CoV-2 and they don't neutralize it. So it's suboptimal neutralization. Right, I mean, sometimes you can actually have antibodies that themselves become receptors and they bind things together, or they enter into cells, or they shield some things that otherwise would be recognized. There are all kinds of strange interactions that can occur. So, so far, the message is that B cells do not seem to be a cure-all in this case. So what about T cells? So what do we know about T cells and SARS-CoV-2? Do they target spike, for example? And there's good evidence that T cells do things that are quite unusual in SARS-CoV-2. So it appears the immunity appears to be both potent and cross-reactive, right? Meaning that you can borrow effective T cell immunity from other coronaviruses. And in CD4 responses are less dominated by targets and spikes than other coronaviruses. It's not entirely clear why. So for example, only about a quarter of all the T cells target spike in SARS-CoV-2 versus about 2/3 in previous coronaviruses. I wouldn't put too much weight on this at the moment because the original coronavirus studies were first done in small samples and two were done a little while ago, so the techniques might simply not be directly comparable. So this would actually require dramatic replication replication for me to really accept it as a significant finding. But it's definitely suggestive. So also pronounced for CD8. So CD4 are helper cells, CD8 are killer cells. So about 1/4 in SARS-CoV-2 versus 1/2 previously. And if you look at convalescent studies, so people have recovered from SARS-CoV-2, their immune system seems to have homed in on multiple parts of the viral genome, which implies that if you want immunization to mimic what a natural infection does, meaning try to make it look as close to a natural infection as possible and lead to as strong a response as possible, then you should probably give the immune system more targets than just spike. And here's a paper that measured exactly where these findings are. So it's a preprint, so it hasn't been published yet, and it's sort of been, it's probably in review somewhere. We don't know that, but eventually will be published. So this was a small study, 28 patients with moderate, severe, or critical COVID-19. So this was done in Germany. And the goal here was to see where the T cell responses go. And these are basically all you need to see, that you have, about 80% of patients had responses to spike, by the same fraction to the matrix protein, and by the same fraction to the nuclear protein. So they're all pretty much the same. So N-reactive and M-reactive means that they, so what you're doing, when you do this experiment, you take a cell, you take, a resting T cell from individual's plasma, and then you incubate it with a particular antigen. So you take a subset of cells and you mix it in with, a viral spike. And in separate reaction, you mix a different subset of the same cells from the same patient with matrix and nuclear protein. So you're basically seeing, do they target something in this protein? So you do all of these, you do basically 3 experiments per individual. So take cells, incubate them with spike, see what fraction reacts, incubate them with matrix, see which fraction reacts, incubate them with nuclear protein, see which fraction reacts. And this is a Venn diagram that shows you that in the 31 individuals, they found responses to all three. And in most individuals, they found responses to at least two. So in fact, there are only 2 individuals that had exclusive responses to spike and nothing else. And these are CD4 cells and these are CD8 cells. So not a whole lot of dramatic differences. And in case you're wondering, these, CD154 plus, CD137 plus, these are the various markers that are displayed in different types of T cells. So this is what they use for flow cytometry to separate the different cells, because you got to know which ones they are. You can't just look at it and say, oh, this is a CD4 cell and it's a CD8 cell. And then just to recap, so this is another way to say what is, I guess, the main message of this paper, which is, T cell and B cell responses are both important, and T cell responses seem to be underappreciated. So recovered patient with mild disease had more prevalent M and N responses than spike responsive CD8s. So again, recovered patients that successfully cleared infections seem to have targets to things other than spike. Patients with milder disease show greater clonal expansion, less active proliferation in CD8 T cells, lower serum cytokine levels. So there's a lot of packed into this. So clonal expansion basically means that you have a couple of cells that are very good at killing SARS-CoV-2. So clonal expansion means they get stimulated and they just pump out thousands or tens of thousands of exact copies of themselves, so they can then go on and clear the infection. And if you have active proliferation, it means the immune system is still looking for a better target. So clonal expansion basically means you have a very, very effective fighting force. All you need to do is just make more copies of an already effective cell. And active proliferation means the immune system has to improve its weapons, right? So it has to develop better targets. And an indication of why T cell immunity is so important for mild disease progression is that if you look at people that had either serious illness or died, they had markers of immune exhaustion. So basically, you had a large number of T cells that express this programmed cell death receptor. So basically means the immune system is overloaded and it's not able to keep up with viral production. So adaptive CD8 T cell immune response in general and broad T cells specifically, specificity in particular, confer protective rather than pathologic effects. Right, and here's another example. So by broad and strong memory T cells, this is a study out of the UK where they looked at 42 patients, and a lot of it is summarized in these charts. So there is so what they did is they looked at mild cases, this category up to the dotted line, and severe cases on the right. Each of the ticks here is a patient represented by a random numeric code. And what you see here is a quantification of how much reactive T cells did they have. So this is the height of the bar. And the colors of the bar is which part of the viral genome did they target. So for example, if you look at patient 11, there seemed to be no T cell responses they could detect. Right, and if you look at, say, patient 06, there was about 1,000 response. Most of it was to purple, which is spike. And there was a little bit to nuclear protein, which is gray, and there's a little bit of matrix. If you look at the severe cases, for example, case 28, you had a much higher response of 3,500 per million cells. So this is sort of a, about 0.4% of all PBMCs were SARS-CoV-2 reactive. And you had a broader representation of what it targeted. So there was a fraction of it going to nuclear protein, a fraction of it going to matrix, a fraction going to ORF7A, ORF3A, and spike. So overall, just by eyeballing this, you can see that one, there was more T cells in the severe case than in mild. So this immune system had to, work harder to deal with this disease. And there seems to be broader response in general in severe disease. So there are more different colors. So then they asked, this is just, but one of the things you could ask is, when the immune system targets a particular protein, does it target just one peptide in it? So does it hone in on just one target or multiple targets? which is what the second plot is for. So this is number of reactive pools, which means they put in different sets of peptides. And if you have a patient serum that reacts to multiple reactive pools, it means that it targets multiple parts of the protein, right? So for example, if you look at patient note 25, in spike, it reacted to 15 different pools, which means it targeted many different epitopes in spike. So this immune system of this individual was really going after the viral spike. And this is basically breadth, right? So you have a lot of different epitopes and a lot of different genomic regions targeted. And I think this is sort of a great summary of it, because you can see that what your immune system is doing is basically going after the entirety of the virus, right? All of its genes and multiple targets within the gene. And it's very rare that you see an individual that is whose T-cell response is dominated by spike. And the reason I keep coming back to spike, and this review kept coming back to spike, is because you had so many vaccines basically placing all of their bets on raising an immune response to just spike. Looking at the other systems, one of the desirable outcomes of a vaccination would be to raise an immunity that stays active for a long, long time. And in order to make the case that this should be possible or might be possible for SARS-CoV-2, the author draws upon yellow fever and smallpox vaccines to provide benchmarks for primary immune response to the viral vaccinations. And these vaccines induce remarkably effective and long-lived T cells. So they're broad specificity, so they target multiple parts of the genome, high levels, polyfunctionality, which in this case means that these are T cells that do all kinds of things that they have to do. So they're both they're involved in cytokine production and signaling, helping, and cell killing. Hyperproliferative potential means that these, if necessary, these cells can be recruited and make many copies of themselves very quickly, and long-term persistence. So in fact, there's a paper, there's this chart that shows you that what they did is they vaccinated people with this yellow fever vaccine. They labeled T cells with a marker that decays over time, so deuterium. It's a radioactive marker that you can basically estimate half-life of. So you see how much of it is left after you've done the labeling. And what they found, that these cells are remarkably short-lived, as they have a half-life of over a year. Right, so the rate of decay is very, very low, which implies that the cells that are raised by the vaccine, that and are observed within a couple of weeks, say a couple of months, maybe 1 month after vaccination, then persist for a long time. And this is what you want a vaccine to do. In human studies of SARS-CoV-2, similar patterns were found, right? So you had in Singapore study that was cited, 100% of recovered patients have broadly reactive nuclear protein-specific T cells. If you look at SARS-CoV-2 patients, all of them had cells reactive to SARS-CoV-1 17 years later. And a lot of them also reacted to SARS-CoV-2 nuclear protein. About half of people that have never been exposed to SARS-CoV-2 had N-specific T cell responses in one study, and 34% S-specific in a different German cohort. The natural SARS-CoV-2 infection broadens S epitope coverage. So what that means is that people that had SARS-CoV-2 and recovered, their immune system targets many different epitopes and spike, whereas if you have pre-existing reactivity, say from other coronaviruses, it tends to go in just one part of the gene. And there were numerous studies that found pre-existing responses, so the US, Sweden, Holland, but there were also some studies that reported that they did not find any. After SARS-CoV-1 or 2 infection, T cells reactive to coronavirus proteins are universally observed, persist for many years, and exhibit cross-reactivity between the two viruses. And one of the interesting points that this paper makes is maybe we can borrow what Jenner did, if you remember, from the smallpox, cowpox vaccine going back to the 18th century, is that there might be some benefit in considering exposing people to mild beta coronavirus infections that cause common colds to basically give them potentially testable protective strategies for COVID-19 prevention, right? So if the cost of having, say, cross-reactive protection against SARS-CoV-2 would be to have a week-long common cold, I would imagine a fair number of people would take that trade. And there's also apparent durability of T-cell responses to SARS-CoV-1, right? So there seems to be 17 years after, we still have a response. Yeah, which is, I think everything is nicely summarized in this figure, which basically shows that you have three major arms of adaptive immunity. So you have B cells, which give rise to antibodies, CD4 cells, which are usually helper cells, and CD8, which are cytotoxic killer cells. So they all start in this naive state, which means that they're looking, you know, they need to be presented with an antigen to which they can adapt. So you can either have acute exposure, which means you actually have the infection, or vaccination. This gives the immune system the viral antigen kick that they need to start proliferating. So proliferating simply means, you know, growing, replicating, and branching off in different lineages. So CD4 cells are involved in all this signaling. So this middle one, helper T cells, are going through, you know, basically involved in all branches of the immune system, adaptive immune system, including, you know, B cell response. But, you know, once you have active B cells, and you have these stimulated antibodies, you can have either viral neutralization or maybe tissue damage in coronavirus, which we don't know. But long term, all we know so far is there's not very good data on other coronaviruses. So there's not a whole lot of memory B cells in other coronaviruses, and they seem to die away pretty quickly. In contrast to this, memory T cells, both CD4 and CD8, seem to persist for many, many years, over 15 years in other coronavirus infections. And there's good evidence that you have these long-lasting memory cells. And if you have these memory cells, which means, what that means is if you have a repeat exposure, you don't have to go through all of this cycle to develop a new response. All you do is basically reactivate existing memory cells, which immediately start replicating themselves, or, you know, CD4 T cells have, you know, to do effector functions. So they basically participate in immune clearance, and then you have CD8 T cells, which take care of the virus, right? So, all right, that's all I got to say about the vaccine at the moment. I thought it was a very sort of interesting paper, and we'll probably, I'll keep my eye out on what else is out there as literature gets updated. We might come back to this, or especially if there's any development in the vaccine front. But to sort of summarize in a minute, all that we've learned so far is, one, there's, you know, 180, probably more, probably 200 now, different vaccine candidates being developed and tested. It's a very broad spectrum. Basically, everything that we've tried before is being tried, and a lot of new approaches that have never been tried before are being tried. What we do see that is not terribly promising is that a lot of the vaccines that are likely to reach the market first are primarily based on antibodies, and a lot of them are based on only looking at one specific antigen, only looking at spike. So if we're being optimistic, based on the information that we have, these vaccines will be effective, but they will not provide either complete protection from infection. So you might still be infected, but you'll probably be asymptomatic, or it might just be harder for you to be infected because they're stimulating IgG, the ones that are delivered intramuscularly. And they might wane. So you might have to give this vaccine periodically, much like we do with the flu vaccine. But for a different reason. For flu, you basically might have a different strain every year. So the vaccine, even if it were effective, wouldn't be necessarily the one that you need for a different strain. But the reason you would need an annual vaccine for SARS-CoV-2 is because the immune response simply doesn't remain. It decays. So we should, as we move forward, and a lot of the vaccines that are in various earlier stages of the pipeline target a broader set of immunogens, and a lot of them might actually trigger the appropriate T cell response. In fact, even if you develop a B cell vaccine with no consideration for T cells, they're probably going to still have some T cell response. Just, it might be too narrow, but we'll see where we are. So, all right, any questions here until we move on to the host factor part? I'll have a all right, seeing none, let's switch gears and focus a little bit on us, the hosts, instead of spending all the time looking at just the virus. And if there's, I'm sure all of you know that COVID-19 does not affect all people equally. There are a large number of factors, host factors, meaning attributes of infected people, that seem to lead to more severe outcomes or death. Just to summarize what all of these factors might be, broadly speaking, so host factor simply means an attribute of a human, an individual. And you can categorize all these attributes into genetic factors, right? So, and this usually means your germline genome, so what you inherited from your parents. You can have environmental factors, which is a very broad category. It involves everything like, do you smoke? Where do you live? Are you subject to, environmental stress? All these things that that are not genetic and encompass pretty much everything else. Demographic factors are, you see them mentioned, but usually demographic factors are a combination of genetic factors. If you're talking about, say, ethnicities defined in terms of genetic markers, for example, or environment. And you also have immune system state, which also doesn't, it's kind of environmental, but it's not quite, because it's, it has a genetic component, but we'll sort of factor it out as a separate. And then if you look at the virus, again, not even though SARS-CoV-2 is pretty genomically homogeneous, you can still have slightly different strains. So potentially there could be some effect whether or not the virus has a particular mutation and spike or doesn't. There's not a whole lot of evidence right now that this makes a whole lot of difference. And then in addition to genetic factors, you might have other factors like, how were you exposed to the virus? Again, not such a big factor for SARS-CoV-2, but it might be for other viruses. So, what are some of the factors that have been implicated as, negative covariates of disease severity? There are numerous papers in this. The one that I have here that was published in Nature in July, but it's really based on an analysis up to May, is a large study out of the UK. One of the sort of unique capabilities that a nationalized healthcare system has, like in the UK, is access to very, very large collections of centralized and standardized health records. So it is possible for them to do a countrywide analysis because they have unified systems. In the United States, it would be effectively impossible to do because all the states and localities would have slightly different regulations, but more importantly, individual hospital systems don't really talk to each other. So, a comparable study in the US might be something that you could do, say, in the VA system, because it's all standardized, or you could do within a specific hospital network, like Kaiser Permanente, where they do standard records. But in this case, it's a very, very large-scale study. So they've analyzed, primary care records of nearly or over 17 million adults. Out of which at the time, about 11,000 were COVID-19 related deaths. Summary of the findings are basically in the abstract. So COVID-19 related deaths with associated with, this is the first keyword that I would like you to focus on, associated with, right? And so it doesn't mean that, being a man, for example, causes more severe COVID-19 outcomes, but it is associated with it, right? So, we'll talk a little bit about these distinctions. And then the report, what this hazard ratio is of 1.6. So in some sense, your odds of dying, if you're a man, all other things being equal, is about 1.6 compared to what it is for a woman. And then 95% confidence interval means that this is another way to estimate statistical significance. So this is sort of, if you've ever done a lab experiment, you measure something and you do replicates and you say, I measured 5 plus minus 2, right? So that kind of gives you a precision estimate. So this is the same thing here. If your 95% confidence interval does not overlap 1, that's statistical significance, but you have some error in estimation. So you have greater age and deprivation, diabetes, severe asthma, and various other medical conditions. Compared with people of white ethnicity, Black and South Asian people were at high risk, even after adjustment for other factors, and I'll tell you what that means. We have quantified a range of clinical factors and so on. So let's look at these data briefly. I think it's important for you to sort of get a better sense of how to analyze these. So this is the cohort that we're working with. So they start with population registered with a general practice using this particular type of software on the 1st of February of 2020, which is 23.6 million. So anything that sort of gets shunted off to the right is excluded from further consideration, which means that if these individuals did not have 1 or more year follow-up, they were not counted. If they were under age of 18, they were not analyzed here. This usually has to do with being able to consent for a particular study. So if you want to involve children, it's a separate layer of patient protection, documentation, and obtaining informed consent. Because in many jurisdictions, you're not able to give an informed consent until you're legally an adult. There are exceptions, of course, that apply, and you can do this, but it requires approvals and special dispensations. So they didn't include children. So if you focus on adults, that's now to 17.4 million, out of which a small fraction had no demographic information or missing demographic information. So it would be, for instance, age, race and ethnicity, and things like this. So they were excluded. So the final included population was about 17.3 million, out of which about 11,000 were COVID-related deaths. When you see these types of association studies, there's a lot of data that goes into this, and some of it will be purely descriptive. So this is, I think I mentioned to you that there's sort of a running joke that when you have a medical paper, there's always table 1. So table 1 is a specific meaning. Table 1 in medical studies is always a descriptive table about who did you study? So what was your study population and how did they break down by various measured categories? And this is a partial table 1. It's actually much longer in the paper. I just included the top of it. So total, 100% of the individuals, were studied. So 0.3 million, out of which, 0.06% were died of COVID. This is the age distribution, right? So this is a population level sample. So this should be fairly representative of just the British population, so about a third between 18 and 39, and 1/6, 40 to 49, and so on, and it tails off as you go to 80 plus. And here's a fraction of those that died. And you already kind of know what to look for. So we know that perhaps the strongest risk factor for dying of COVID is being of advanced age. And you can see this already from this table. So and what you need to see is look at the parenthetical numbers in the second column. So if you're 18 to 39, your probability of dying within that age group is basically 0. 40 to 49 is 0, and then it starts ticking up. So it's now 0.02, 50 to 59, and goes all the way up to 0.57 if you're 80 plus. So this is a dramatic difference, right? It's obvious to the eye, and you should be able to see it by statistical analysis very simply. It's like, if you go back to my example, it's having, the two bins of data that are very far apart. And if you look at sex, male or female, so again, it's about 50-50 because it's a population level sample, and you expect, in most societies, the male to female ratio to be close to 1-to-1. And I already told you in the abstract that they found a significant difference, and you should see it here. So it's about 6%, 0.06 within this stratum, and 0.07 if you're male. So it's different, but it's a lot less different than you would see by age, right? So it's statistically significant, but it's not so obvious to the eye. And then as you go, so, for this, you need to run some statistical tests, to convince yourself that it's true. Then the next category is a measure of obesity. So BMI stratified. So this is considered to be the healthy range. And then you go into obese class 1, 2, and 3. And you can see, there's a little bit of a gradient, not great. I mean, you're a little more likely to die if you're in the obese class as opposed to this. But then if you're also, under 18.5, so you're below what BMI considers to be healthy, which is probably in the old and frail category, then your chance of dying significantly increases. Smoking, ethnicity, and so on. So this is just a description of the data. When papers like this present statistical analysis, they will take the same categories, but they will now represent them in something that's directly interpretable. So let me see if I can, and this is sort of a relative risk analysis. So let's spend a minute or two on the slide. So for instance, you want to look at the effective age group. All right, so you have 1, 2, 3, 4, 5, 6 age groups, and you want to see if there is a different risk of dying from COVID if you have different age groups. So you designate one of these risk groups as a reference, right? So, this is just a part of the statistical analysis. So here, and you can arbitrarily choose which one it is. So here they pick 50 to 59 to be the reference. So all these other statements are relative to people that are between 50 and 59. There's a question how you decide on the reference category. You don't, a lot of it is arbitrary. One of the things you don't want to do, you don't want to pick a category that is very small, just because you can have, numerical stability issues, but by and large, it's arbitrary. You just pick one of them as the reference level. So the way to read this is, there is your reference risk is, if you're between 50 and 59. If you're younger than 50 or 59, you have this hazard ratio, which is, again, is basically a relative risk. So loosely speaking, you're, a third as likely to die from COVID if you're 40 to 49 as you're 50 to 59. And then it's adjusted for age and sex, which means that when you run this analysis, you also consider men and women separately. So this is not confounded with whether or not you're male or female. But this is also a really nice gradient, and you can directly read this off. So your relative risk, for instance, or hazard ratio, if you're 80 plus, is over 40 times what it is if you're 50 to 59. And if you're in the 18 to 39, it's 1/20th of what it is if you're 50 to 59. And fully adjusted means you do an analysis where all these variables are put into the model. So the effect will always be smaller if you do fully adjusted. So all the things are going to move closer to 1, but they're still significant. And you can see what it is for male and female. You can see what it is for obesity, right? So these are all significant, but with a much lesser effect. And you can also see some of the things for which you shouldn't, you might imagine that if you smoked, for instance, there's a good reason to have prior belief that smokers should be at higher risk for dying from from pulmonary diseases in the non-smokers, which is what you see, that older people are more susceptible, that obesity correlates with a number of adverse health outcomes. But for instance, if you look at ethnicity, there's no real reason to believe a priori that any ethnicity would be more or less affected than any other ethnicity. But in this case, it seems that if you take white as the reference, then every other ethnicity that's considered in this study has a higher hazard ratio. We'll come back to this when we talk about various ethical considerations of how do you explain this. And there are a lot of confounders that go into this. So one of the standard explanation is sort of health disparities. That people from minority groups, just tend generally to do more poorly with health-related outcomes because they have less access to quality care, poor insurance coverage, and many other things like this. And then IMD quintile is basically a measure of what they call social deprivation, right? So this is, basically a measure of how many non-health related stressors you have in your life. So for instance, are you unemployed? Do you have substance abuse issues? Or, do you suffer from, lack of food access? So things like this. And here you also observe an expected gradient. So higher degrees of deprivation correlate with poor COVID-19 outcomes. Question is, why are white people generally considered control groups in most research studies? It's a good question. So I don't have a universal answer for it. I mean, a lot of it is historical, and for well-known reasons that just racial inequalities in general. But it's sort of recently there's been a lot of effort, at least on part of the federal research agencies in the United States, to correct for such disparities. So you have, especially if you have indication that a disease disproportionately affects people of a different ethnicity, you will put more effort into studying this. But in this particular case, they picked white as a arbitrary reference. You could pick others, but it also makes a clean interpretation because everybody else is at a higher risk. But by and large, for most, if a disease affects, there are some diseases that affect different ethnicities differentially because of genomic markers that come with these ethnicities. And they just randomly have to do something with a particular gene that has a specific function in disease. But in this case, there's no a priori reason to believe that coronaviruses should affect people differently. Although we see that men and women are affected differently, and that's still not fully explained. I might tell you next time one example of how there's one small explanation for why men are more susceptible. And if I were to tell you what it is now, is if you have, the genomic differences between men and women is that we have 23 autosomes and we have X and Y. So women have two X's and men have X and a Y. So if you have anything that's X recessive, so if you have any feature that requires to have, that is only expressed if you have two broken copies of X or only one copy of X, that'll affect men more than women because women will have another copy of X to potentially compensate for it. So I'll show you examples going forward. That's all we have at the moment. I'll throw up a paper that I'll come back to next week because we need to do a little bit of background in order to get to this. So there were two very interesting papers published in Science back to back from the same research consortium that looked at two different genetic bases of disease. Specifically, the one paper that I cited here, entitled "Inborn Errors of Type 1 Interferon Immunity in Patients with Life-Threatening COVID-19," so the object of this paper was to study if people have rare mutations in genes that are responsible for innate immune response, do some of them have a worse outcome with COVID-19? And the answer to this paper is yes, but before I can explain to you why, we have to do the rest of today's lecture with just background and terminology, and to talk to you as well about something other than COVID-19. Switching gears a little bit, we can broaden our perspective to look at genetic bases of all types of diseases, not necessarily viral diseases, but all diseases you might imagine. So acute diseases, chronic diseases, things like diabetes, heart disease, risk of cancer. And if you don't know anything about a particular disease, it is safe to say that what causes this disease is going to be a combination of many factors. The vast majority of diseases, some of those factors are genetic. So when I talk about genetic basis of disease, I talk about the effect of specific mutations in the human genome, in this case, that could either occur in coding or non-coding regions, so either within a gene or outside a gene, that can give rise to a disease, which means that the genotype of an individual might affect the disease phenotype. So do you get this disease? Are you more likely to get this disease? If you do get it, is it going to be milder or more severe? And the dependence between genotype and phenotype can broadly be broken down into these three categories. It can be monogenic. Mono means one, so one change explains most of the phenotype. Oligogenic means that a few changes explain most of the phenotype. Polygenic, which means you need a lot of different changes that put them together. When you put them together, explain the disease phenotype. When we talk about genetic basis of disease, the implication is that it must be heritable. So it must be something that you received from your parents. Not 100% true, but close to 100% true. So some of the disease phenotypes that we could be talking about in this case, they span the entire spectrum of severity. So perhaps the most dramatic disease phenotype would be something like a birth defect, which is some of the first diseases that people were able to study because they were obvious to diagnose and they were fairly rare. And also coincidentally, they seem to have a very simple genetic basis. You can have a lifelong condition, right? That is not, a visible or otherwise adverse effect, but you could still have something like, a good example would be something like myopia, right? So short-sightedness. It has a genetic basis, not a simple one, and it is a lifelong condition. You can also have a disease phenotype that is not an actual disease, but simply an increased risk of developing a condition. So a good example of this would be BRCA1, BRCA2 gene mutations, which predispose, mostly women, but men to a lesser degree as well, to a much greater chance of developing breast cancer. You could also have a carrier phenotype, which means that you have a mutation that you carry, but it doesn't manifest in you, right? You can pass it on to your children. So this is like the silent carrier. When you talk about disease phenotype, one of the terms you will see thrown around is penetrance or effect size. And it's defined as the likelihood that a person carrying a disease-associated genotype will develop the disease phenotype. So strong penetrance or high or large penetrance means that if you have this mutation, you're pretty much guaranteed to have the disease. And low penetrance means that only a very small fraction of people that have a particular mutation will develop the disease. So some of the simplest diseases, and actually, if you go to the science paper that has to do with SARS-CoV-2 and interferon deficiencies is genetically very simple. And it deals with so-called Mendelian diseases. So Mendelian diseases are called so because they follow the Mendelian inheritance pattern, which means you can explain their inheritance patterns through the evolution of a single locus. And they're called monogenic diseases. They sometimes run in the families. They were also the earliest form of genetic studies, the study of hereditary disease. So there are a number of examples, but perhaps, the one that always pops into my head in this context would be, hemophiliac disease in various European royal families. It's a classic hereditary disease there. So typically, these monogenic diseases are caused by mutation in a specific locus, which means specific gene, but with a very strong effect or high penetrance. And the environmental contributions to these are very, very minimal. Question is, what about the level of expressivity? Does it also apply in this case? You mean like regulatory changes? So, mutations. Yeah. So it does apply. So these are also genetic changes, and then they can also have Mendelian inheritance patterns, but they don't change the function of the gene. They change how much of a gene is made. Yeah. So these are very, very common. In fact, most of the disease-associated variants that we know of are so-called QTLs, so EQTLs, so expression type loci. So they do something, but they don't change the function of the gene. So what are some of the examples of classical genetic diseases? So the one that I always like to start with, because it's one of the things, it's not necessarily a deadly disease, and it's one of the best studied ones, and it kind of has an interesting twist about malaria and adaptation, is sickle cell anemia. It has a very simple genetic basis. It is caused by a single mutation. It's what's called by point mutation. So literally one nucleotide changing amino acid in the beta globin gene. So the globin gene family, so you have alphas, betas, gammas, I forget how far they go. I think they're epsilons. And so they all tend to do with red blood cells and oxygen transport. So beta globins and alpha globins, they form up together to produce red blood cells, which, so if you look at this inset picture A here, the typical red blood cell looks like a disc with a dimple in the middle. And the dimple in the middle is where the oxygen binds to. They also have these nice pliable round shapes, which means that they can go around bends and blood vessels and not get stuck. So this dimple is an important thing because it allows efficient oxygen transport, and the shape and flexibility allows the cell to get around in very thin blood vessels. If you have this mutation, and it's not a unique mutation, but it's a mutation in the beta globin gene, you have a malformed protein. So you have two normal alpha hemoglobin units and two mutant beta globin units form something called hemoglobin S. And it has a very simple phenotype. So you can actually see the change in the shape of the cell from this nice dimple to something that actually looks like a sickle, which is why it's called that. Right? So it looks, if you imagine a handle here, that'll look like a sickle. So if you look at this cell, there are two obvious things. They actually aren't able to carry oxygen as well, and they also stick together and can block blood vessels. So in addition to these morphological changes, they also have shorter lifespans. So normal blood cells, live about 3 to 4 months, and these sickle-shaped cells only live a couple of weeks. From the standpoint of how this manifests itself in an individual that has this phenotype, so typically manifests itself in low oxygen or high stress conditions. So you have lower elasticity and oxygen carrying capacity. So you will basically, run out of breath, not be able to exert yourself so much. And this will become obvious if you, go into the conditions where you're required to tax your oxygen transport systems. The question is, why is it that sickle cell anemia confers an advantage to malaria resistance? First, it's a great point. I actually have a line, I think 2 slides from now, to indicate this specifically. But one of the reasons, so malaria, Plasmodium falciparum, which is the most common parasite that causes malaria. It's not the only one. There are other related ones. But it has a very, very bizarre, complicated life cycle. One of the stages of this life cycle involves red blood cells. And if you have sickle cell misshapen blood cells, then it really makes it an inhospitable environment to the malaria parasite for this life cycle. So it's simply not able to complete one of its life cycles because the cell that it normally replicates in is malformed and it's not able to do it as efficiently or at all. It's a classical Mendelian trait. So it's called an autorecessive or autosomal recessive and codominant. So autorecessive means, so autosomal means it's not in the sex chromosome, one of the normal chromosomes that you have, a pair of homologous chromosomes in both men and women. Codominant means that heterozygous, when you mean you have a normal protein on one chromosome and a mutated protein on another chromosome, means that you make both healthy and unhealthy blood cells. Is acquired from two carrier parents who are asymptomatic, right? Obviously, if you have a symptomatic parent, that's a different pattern of inheritance. But here it's shown in case neither parent is symptomatic. And the heterozygous form has a selective advantage against malaria because the parasite is not able to replicate as efficiently, which means that to this day, it maintains relatively high frequency in Africa, where malaria in some areas is endemic. As an interesting example of natural selection in action, among Americans of African descent who left the African continent, potentially hundreds of years ago, moved to an environment where malaria is not a thing, or at least it hasn't been for quite some time. What you can see is this selective advantage is lost. So the frequency of the heterozygous form has been significantly diminished over time. So in the US, amongst Americans of African descent, it's only about 0.25%, whereas in African populations, it's about 4%. We'll be talking about different inheritance patterns for the SARS-CoV-2 paper and elsewhere. So I'm sure you've seen this all before, but just to remind you, these are really neat, much better pictures than what I remember from my genetics textbooks when I was a kid. So this is all courtesy Wikipedia. So you have, a cartoonish representation of this allele, where you have dark blue meaning wild type and light blue meaning mutant allele. You have a carrier father, which means that it's a heterozygous variant, healthy, a normal wild type and a mutant, wild type and a mutant. You have a traditional Mendelian inheritance pattern because it's autosomal, doesn't make any difference if the child is male or female. And then you get to have 4 combinations. You pick one from the father, one from the mother. These are all the 4 combinations. In one case, you'll have a child that, 1 out of 4, both wild types. So this is unaffected child. 1 out of 4, you'll have a child that has both affected variants. So this is now homozygous, so disease manifestation. And you have a carrier child that doesn't manifest the disease because, you're making healthy protein in addition to some malformed protein, but both of them are carriers. So autosomal recessive single gene disease occurs only in homozygous individuals. Those who carry 2 mutant alleles with the disease-associated types, and heterozygotes are silent carriers. So another classic example of this, is one of the diseases of the early 20th century when it was characterized, and it was sort of one of the many diseases that now form the group of standard tests that every baby born in the United States undergoes. So they are neonatal tests. So PKU, or phenylketonuria, is also another autosomal recessive disease. It's completely different in what it does from sickle cell anemia, but it has a very similar genetic basis. It's associated with mutations in one gene that encodes one enzyme called phenylalanine hydroxylase, or PAH. And it's one of the many diseases that have to do with metabolic or amino acid processing issues. So when a person has these mutations, they're unable to break down phenylalanine, which is one of the 20 amino acids, which is an essential building block of dietary protein. So, if you start with phenylalanine, you have two metabolic pathways. So one of them is to make tyrosine, which is another amino acid. And that requires the presence and action of PAH, so phenylalanine hydroxylase. Through a different a chemical pathway, through the action of a transaminase, you make a different compound, which is very similar to tyrosine. It's called phenylpyruvic acid. So if you have a broken balance between these two reactions because this enzyme isn't functioning, you make too much phenylpyruvic acid. If nothing else is done, it builds up over time. And once its concentrations reach a critical level, it can become toxic to brain development. And if left completely untreated, it will lead to things like mental retardation. The brief history of this disease is actually shared by many diseases of the same class that were discovered, about 100 to 150 years ago. So this particular one was discovered by the Norwegian physician Ivar Folling in 1934. And the way this was discovered is sort of very characteristic of early, pre-sequencing genomics. Is one, you have to identify an easily associated condition. So you should be able to diagnose individuals that have the particular condition, so you can try to analyze what's common between them. So he observed that this particular condition, hyperphenylalaninemia, alaninemia, HPA, so having too much phenylalanine, was associated with intellectual disability. Right, that's not particularly diagnostic because there are many different conditions that are associated with intellectual disability. However, he also noticed something that is easily detectable, which is this: the urine of affected children has a very characteristic smell. Which he was then able, by the analysis of said urine, trace the origin of the smell to the overpresence of phenylpyruvic acid, confirmed in multiple other samples. And basically now you have a chain that having too much of this acid leads to intellectual disability with hyperphenylalaninemia. I'm not going to try to pronounce it, HPA. So in the 60s, there was a sort of first neonatal heel prick test developed by Robert Guthrie, introduced the newborn screening test in the 1960s. And this is sort of a seminal moment in genetic disease diagnostics, for the reason that if you're able to very early in the child's life realize that they suffer from PKU, then you can treat them immediately and basically get rid of all the side effects. But you have to do it very early, and you have to do it before any of the ill effects accumulate. So an affected newborn can grow up with normal brain development through diet or combination of diet and medication. So for instance, one of the simple things you can do is just alter your diet. So eliminate foods that are high in phenylalanine, so things like egg whites, shrimp, chicken breast, nuts, and so on. And then you can supplement your diet with protein substitute formulas, because you'll also have a deficiency in tyrosine. And you can replace it with supplementation. And this was a sort of bacterial growth type test where you take a bacterium, in this case Bacillus subtilis, that was incubated in an agar gel in the presence of an antagonist of phenylalanine, which prevents the bacteria from growing. If you then take paper discs that are soaked with blood that have too much phenylalanine, this phenylalanine will overcome the action of the antagonist and promote bacterial growth. So a positive readout of this test would be like this XV here, where you have a bacterial colony. And it's something that you could do this in an industrial scale. It's a simple test, easy to run, easy to read. It's obviously done differently now, but this is how it was done in the 60s. So depending on which state you're in, if you have a child in different states, they will undergo sort of a different battery of tests. I use California here mostly because they have sort of the most easily accessible statistics. Their equivalent lists for Pennsylvania or any other state that you're interested in doing, they all have some form of newborn screening. But here's some of the conditions that the state of California screens for people in California newborn screening program. And here's the number of cases that they diagnosed over the life of the program. So for example, classical phenylketonuria it's a rare disease, so the prevalence is about 1 in 40,000, but a fair number of babies are diagnosed with it annually, so 15 to 18. Other rare conditions like classical galactosemia, congenital hypothyroidism, sickle cell disease, which see much more prevalence than PKU, cystic fibrosis, so on. So we're talking about hundreds of individuals that are diagnosed early. And for many of these conditions, if you deploy the right treatment right away, the condition is pretty much cured. Not for all of them. Cystic fibrosis, for instance, isn't, or sickle cell disease itself is not curable. It's manageable, but it's not curable. So here's some of the examples, sort of just four examples of monogenic autosomal recessive diseases. So where you talked about phenylketonuria, so overabundance of phenylalanine. It affects the gene called phenylalanine hydroxylase. It is treatable through diet and supplements, and early detection is critical. The prevalence in the US is between 1 in 10 and 1 in 15,000. So cystic fibrosis is a disease of quote unquote thick secretions. It's caused by sort of primarily by mutations in this gene called CFTR, cystic fibrosis conductance regulator. It's the disease itself is not treatable, but you can manage the symptoms and have a fairly long quality life, so 45 to 50 years life expectancy. One of the interesting things that you will notice about this disease is that it affects different ethnicities differently. So for instance, this disease happens to be the most common among Caucasians, and that's about half as common among Hispanics, and about only a quarter as common among African Americans. And you can go and try to find the markers that are associated with it and people have. But it's actually quite common for genetic diseases that it affects different populations differently. And that simply has to do with the fact that different populations have different histories, migration, and so on. So sickle cell anemia, beta hemoglobin, again, symptoms management, over 50% live to be 50 or older. This one is much more prevalent among African-American infants, so about 1 in 400 versus 1 in 4,000 overall. You have some other conditions like ocular cutaneous albinism, decreased or absent pigmentation in the eye, so and the skin, so symptom management, special skin and vision care, and so on. So now with that out of the way, I'm just going to briefly go through some of the other diseases that have different inheritance patterns. So most of the diseases that you're likely to see in the lists of genetic diseases, or the ones that are commonly screened for, are autorecessive. There are a number of important diseases that have a different inheritance pattern. So they're autodominant, so autosomal dominant, which means that all you need is one mutant allele to have the condition. So one carrier parent is enough. And it's a mutation in the HTT gene called Huntington. If I'm not mistaken, and I haven't checked in the last year or two, that it has this is a complex gene that hasn't been fully functionalized and fully functionally characterized, and it has a mutational pattern different from sickle cell anemia. So instead of just changing one nucleotide, this gene adds some trinucleotide repeats. So it just adds more nucleotides, and the more you add, the more severe the disease is. It generally results in the death of brain cells. The inheritance pattern for this disease is different because, and that's here it's indicated that you have an affected father. Doesn't make any difference. The same pattern would occur if you had an unaffected father and affected mother. So here you have basically half of the children are affected, the ones that get this allele from the father, and half the children are unaffected. Doesn't affect genders differently because it's autosomal. But if you remember, in the autosomal recessive, you had two carriers, one unaffected, the one affected. Here you have half unaffected and half affected and no carriers. I mean, I suppose carriers there's no carrier status. So some of the examples of autosomal, sorry, is this? No, I think, I meant to say this is the dominant one, right? So, just examples of, mutations in different genes, whether or not treatable or not, and prevalence that has been estimated. Now, a different pattern of inheritance occurs if you're looking at sex chromosome linked diseases. So Duchenne muscular dystrophy is probably the most common X-linked disease. So it has an X-linked recessive monogenic disorder. So generally men are affected, women are carriers. The symptoms are muscle weakness in childhood, which then leads to progressive loss of function, cardiomyopathy, and eventually death. Between 400 to 600 newborn boys in the US have this condition each year. Females can develop the condition, but it's much rarer, typically milder than present. So it's a recessive monogenic disorder, which means you can you will get it if you don't have a healthy copy of the X. So in this case, women have an extra layer of protection. They have two copies of X. So if one of them is healthy and the other is not, then the child is unaffected. If the affected son inherits the copy of X from this case carrier mother, is going to be affected. So cardiomyopathy, as a question, what is cardiomyopathy? Cardiomyopathy is sort of a constellation of diseases that have to do with the degeneration of the heart muscle. It basically loss of function, right? So your muscle weakens or becomes too hard or is not able to contract the heart, and this sort of eventually leads to heart failure if untreated and sufficiently severe. But it's just basically any abnormality in the heart muscle, usually of degenerative kind. So in here, right. You also have X chromosome linked dominant monogenic disorders, so things like Rett syndrome, a rare neurological disorder caused by mutations in this gene. This is sort of a master regulator gene, so it's involved transcriptional silencing and regulation of methylated DNA. It's rare, occurs in 1 in 10,000 female births. And interestingly enough, this is sort of a strange disease in the sense that of 95% of cases, the syndrome was caused by de novo mutation in the child, in the sense that they can't find mutations in this gene in the parents. So it just occurred randomly during meiosis or mitosis. And you have different inheritance pattern based on whether or not you have affected mother or father, right? So here, the only way you don't get this is if you do not have any copies of the broken X. And it can happen two ways. Like in this particular inheritance pattern, the unaffected son gets his the healthy copy of X from the mom and the Y, and in this case, the affected son will get the mutated copy of X from the mom and the Y from the dad, and same pattern, a different pattern, but same outcome for daughters. If you have an affected father, the inheritance pattern is different, which basically means that affected father, if you have a son, they will be unaffected because by definition they cannot inherit X from their father, but the daughters will deterministically have the disease because they have to inherit one X from the father. And here's, you can also have Y chromosome linked monogenic disorders, which only deterministically affect men for obvious reasons. Since women do not have a Y chromosome, these conditions are rare. The question is, for these genetic disorders, you could get gene therapy, or has this not been approved by the FDA? I'm not 100% sure on the status of gene therapy. I know some of gene therapy treatments have been approved, but I'm not prepared to tell you which ones. We can find out, um, and it's definitely being actively researched. So yes, you can. Gene therapy is basically, it's a mechanism where you can either edit uh, a mutation in the germline, or you could try to deliver a functional protein or a functional gene somewhere else. And it's definitely a very, very promising area of research, but it's still fairly new. So what I would like to you point out here is that in addition to inheriting one of the potentially disease-causing variants from your parents, there's a small but significant probability of generating de novo mutations, which means that these mutations are unique to you. So they're present neither in your mom nor your dad, and they just occurred randomly, or when the sperm and the egg were going through the early stages of fusing and replicating. And the orders of magnitudes are very, very different. So here's an interesting table that if you look at all different types of variants and different types of attributes of the variants, if you look at inherited variants, there's about 3.5 to 4.4 million per individual. So what that means is that if you pick two random people that are not related to one another, in the recent past, and compare their genomes, you will find about this many differences. Or to be more precise, you will find probably twice as many differences, because this comparison is relative to the standard genome. And if you think about it, there's 3.5 million variants. Our genome is about 3.5 billion bases, which means that any two people in this class, unless you're related in the last couple of generations, are different at about 1 in 1,000 positions in our genome. Every individual, on average, gets a much smaller number of de novo mutations that are unique to them. So somewhere in the order of 44 to 82. If you start breaking them down into what type of variants there are, so there's a single nucleotide variant. This is about 22,000 in an average individual compared to the reference genome versus 1 and 2. You could have short insertions and deletions, long insertion and deletions, copy number variation, which means you get an extra copy of the gene, exists, very rare. Selection pressure in previous generation, high, right? Because if you happen to get a deleterious mutation, it's going to be at a very strong selective pressure not to move along. By definition, de novo mutations cannot be selected because they were just generated. Damaging capacitive variants. If it's a mutation that's present in many individuals, probably small effect. There's a high chance that a random mutation can have some damaging effect. Parental paternal age effect. So this is probably the most striking difference between them. Inherited variants do not increase if the parents are older, but de novo variants are found at higher rates if the mother or the father are older when the child is born. And de novo mutations are a major cause of severe early onset genetic disorders, so things like intellectual disability, autism spectrum disorders, and other developmental diseases. And the occurrence of novel mutations in each generation might explain why these reproductively lethal disorders can continue to occur in our population. Right? So if you have, imagine that there's a particular disease that sterilizes the individual, right? So they're not able to have children. By definition, they're not able to pass it on to the next generation. But if the disease keeps occurring, then the only way it can occur if it's generated randomly in a very small number of individuals by mutation. And de novo mutations are predominantly of paternal origin and increase with advanced paternal age. So this is something you might not have known about. So there's a well-known increased risk of certain genetic disorders with the age of the mother, right? So Down syndrome, for example, has a very pronounced increase in occurrence in mothers who are over 40 years old, and children born to mothers that are over 40 years old. But de novo mutations seem to be occurring at a higher rate if the father is of an advanced age. And the common wisdom says that there's really no difference at what age you have a child if you're a man, but it's not quite true. All right, so I'll skip this, and I'll conclude by just showing you a really neat database. So let me stop recording here.